Good morning, everybody. Today is Thursday, July 28th, 2016, and this is Before the First Cup with Jules here on UCY.TV. Happy Thursday, everyone. It is 67 degrees and partly cloudy here in upstate New York. Looks like there might be a little bit of rain on the horizon for the next few days in the upper northeast, so that's always a good thing. So where do we leave off yesterday? We titled the show Gates, Oxitec, Zika, Congress, $1 billion, tips, DNA, and DARPA. And yeah, that was a mouthful. But we did go with all of those things. Good old Billy Gates sticking his toe into all kinds of this new mad science gone mad technology. And all of it seems to have a lot to do with modifying genes and being able to take control of the building blocks of life, being able to manipulate and identify all of the different components that make biology, and then be able to create your own version of that biology. And we've been seeing that time and time again. There has been quite a spate of scientific studies and experiments going on right now. We've looked at the CRISPRing humans quite a number of times. First, it was just the egg and in a petri dish for some embryos that would not be allowed to grow to term. I think it was only a week or so that they were going to allow them to grow in the U.S. and in the UK, as well as in China. And now we are seeing that they are having a race to see who is going to be the very first to CRISPR humans. Both experiments, one at Penn University and one in China, having to do with modifying the immune system, T cells, white blood cells, and have them be modified to identify certain types of cancer once they are infused back into the body and be little search and destroy machines. Pretty scary when you think about it, because like we've said a million times, there's always a double-edged sword, and if it can be used to attack certain types of cells, let's say, and go after them, this being cancer, which is the silver bullet, why couldn't they be modified to go after other types of cells or be programmed to release some sort of toxin at some point in the future? And we saw right at the end of the show, we were looking at E. coli being able to be programmed to kill cancers, and that was by programming the cells produce chemicals that then are released into the bloodstream near the tumor and kill it. So, like I said, who's to say we couldn't have an engineer some arsenic or whatever on cue or after a certain period of time for an expiration date on that biological entity that couldn't be traced back, let's say, to some sort of external influence. I want to talk about life forms in general. I saw a really messed up story the other day out of ScienceAlert.com titled, "Researchers have discovered a new life form in our saliva, and it is linked to human disease." Now, really makes me wonder, if you remember, we went back to Dr. Huber long time ago when he first started talking about glyphosate and some of the damage that it was doing to the livestock and to the crops, and some of the things that he was finding when he was inspecting the soils and the pathology of what was happening and why these animals were dying. We had stillbirth abortions and non-productive crops. And Dr. Huber had identified an unidentified life form, he called it, specifically, in the soil that seemed to be there after the glyphosate killed the beneficial bacteria, because glyphosate is a potent antibiotic. And then shortly thereafter, going through the Dr. Huber interview on the show, we took a look at another lecture. This one was by Sophia Smallstrom, who we've heard interviewed quite a number of times by different hosts that we air here on UCY. And Sophia is very intelligent. I really like listening to her. She's got a lot of great connections that she's made between a lot of these different synthetic biology experiments that have gone live in our environment. And one of the things that she was showing was that they were taking air samples, and I don't remember if it was of the spider webs. It might have been the spider webs that they've been finding in the air. Some of those little tufted things that come down out of the sky. But she had some analyses done of things that she'd pulled out of the atmosphere. And one of the things that she found was that we had different branches of life that somehow now had combined and included red blood cells and all kinds of strange things, but had combined now into something that looked like a new life form. And I had connected that dot at the time with some of the things that Dr. Huber had been talking about in regards to what he was finding in the soil as well. So we have the soil in the air, but now this story comes out. Researchers have discovered a new life form in our saliva, and it is linked to human disease. And what really struck me about this is, in our saliva, first thing I would think of is we are putting something in our mouth that caused this new life form to take hold in our saliva. And one of the things that would go into our mouth would be, crops grown in soil where Dr. Huber may have found this new life form. And what else in our mouth is air? So it's just very strange to me. Talk about let's call it a green dot, not black, not with a black Sharpie line yet anyway, but a green little dot to be like something to research, because I thought this was very odd. And I always told you, if there's something that you read and it makes you raise an eyebrow and go, huh? It's something you should definitely pay attention to. So let's read this story to kick this off. Researchers in the U.S. have discovered a brand new life form hiding inside of our saliva. It's a type of parasitic bacteria that requires on other bacterium to live. And I had read a story that must have been the same thing, but written from a different perspective, where they were saying that they found bacteria living on top of bacteria inside of bodies. Well, when we dug into the microbiome and gut bacteria, we discovered a long time ago through the vagus nerve and all these different control capabilities that our gut has over our brain, that gut bacteria was not only important to our immune system, but it's also extremely important to our mental health. And when our bacteria is damaged, we then have bad bacteria that starts taking over, and it causes inflammation, illness, and disease, as well as depression and many other things. So they have been now really digging into the microbiome to understand how important it is to our daily lives. We have companies such as that Synlogics, that is partially financed by some Bill and Melinda Gates money out of MIT, that's spun into its own company now, that is going to be building synthetic biotics as a quote-unquote drug to cure your illnesses. That MIT Synlogics was making synthetic gut bacteria because, see, they understand how important that is to your overall health. But remember, this big push for understanding the brain and mental health is going to be massive as well. They can, through push buttons that create certain chemicals in the brain, manipulate living creatures. They've been experimenting with push-button chemical creation in the brain for a long time through DARPA and some of these brain experiments that they've been doing. And we've done countless shows on the brain, different experiments of part of that brain initiative that's been going on. And it's not just at DARPA, it's at all the big universities and the hospitals. Johns Hopkins University has the biggest database of children's brains to boot. Anyway, so back to this new life form now that they're finding in our saliva. Talk about absolute insanity, never before seen, right? This is probably the biggest one yet. So it's a type of parasitic bacteria that lives on other bacteria that happens to be living in our saliva, and this bacteria only has 700 genes and is the first strain ever discovered that is completely dependent on other bacteria for survival. And if that wasn't creepy enough, this new life form has also been linked to a whole range of human diseases, including gum disease, cystic fibrosis, and antimicrobial resistance. This new parasite, which is yet to be formally named, so it is hot off the presses, wink, out of the lab. I mean, what? This is just insanity. So all these years, all this shit, we just discovered a brand new life form living in our mouths. I don't see how that's possible. Maybe, but I really don't see how that is possible. Out of the blue, never before seen. It was discovered by scientists at the University of Washington School of Dentistry on a mysterious fragment of RNA in human saliva test that did not match any known organisms. Other labs found the same strange DNA code in the past, but no one had been able to figure out where it came from, so it's been around for a while. But after studying it more closely, the team was able to show that this RNA belonged to tiny bacterium that's actually living on another, better known strain of bacteria, and that is the Actomyces adelolictius. I think I'll spell it Actinomyces. Second word Odontolyticus. Although scientists have found bacteria that can infect other bacteria in the past, this is the first time that they've ever discovered a strain that is entirely dependent on infecting its host because it cannot make its own amino acids that are essential for life. So it's like a total vampire. It sucks another one's life dry like a psychopath. It's the psychopath bacteria. I hope they name it that. And that is why it was so hard for scientists to identify it, because they cannot grow it and keep it alive in the lab without its host. So it's like some sort of weird-ass symbiote. You know, one of the other stories I was reading is that our body, right, we did a story on the 5 different colonies of bacteria we have on our body. I can always only remember 4. There's nasal, there's oral, there's skin. Oh, I guess so. There's intestinal and there's vaginal, so it's 5 on females. I guess 4 on males. But those different colonies of bacteria all have a symbiosis between each other, and not necessarily like this where they suck off of each other, but they all live in harmony or discord, and they said that they can even have battles, on your body, but each different one influences different bodily processes in humans and other living biological creatures. And they said that we have, out of our, I think it was like 1 trillion cells or something in the human body that they're trying to sequence right now and understand. Maybe that was the DNA. But we have bazillions of more bacteria cells on our body than we do human cells. And that's the big deal, because we are actually being manipulated and controlled by the bacteria in our body. When we read about that toxoplasmosis, there were a couple different things now, right? That was putting the love goggles on to love your kitty cat. They had found that toxoplasmosis when mice were infected with it, that it caused the mice not to have their natural fear that should be bred into them of predators. They decided that they loved the kitty cat and they'd go rub on them and stuff and to make it easy for the kitty cat to eat him. But they were thinking that actually came about, that toxoplasmosis relationship between the mouse and the cat came about through an evolutionary process, and that the bacteria was actually the control structure that learned to infect other organisms as a survival mechanism by then providing its host with sustenance. And not only can toxoplasmosis infect humans and give them kitty love goggles, but it also can give them that rage disorder they're discovering now. I've seen a couple different stories on that. But it's disturbing to think that all this time we thought we were this, living biological entity that had our own free will, and we discover that there's all these other little microscopic life forms living inside of us that might actually be pushing the buttons and controlling us like a marionette. I mean, that really freaks me out. That's almost like Invasion of the Body Snatchers, and we didn't quite know it. We'd always been snatched, All right, anyway, back to this foreign life form living in our mouths. So they require their host, and they don't make their own amino acids, so they are symbiotic or parasitic, let's say, not symbiotic, parasitic to their host in order to survive. They are ultra small bacteria, and they live on the surface of other bacteria, says lead researcher Jeff McLean to the annual meeting of the American Society for Microbiology in Boston on the first week of July. By studying this parasite in the lab alongside its host, the team has now shown that the new type of bacteria hunts down actomyces and attaches to its outer membrane. And at first, the actomyces can handle the parasites which suck the nutrients out of it, so they are the parasites. But eventually it comes, and it is killed by the attack, so that's not good. Killing your host. You're supposed to only suck a little bit and then let it grow back. That's a problem. Some narcissists do that too. They'll kill their host by just keep sucking them dry. See, they don't understand that, because instead of sucking them dry, chucking them, and trying to find a new one, that's pretty difficult. That's like trying to get a new employee constantly and doing all the retraining. It doesn't quite work out that way. You should always get your victim to have a slow death, because it's much, much more profitable. And, that's kind of what the New World Order does to us. It's slow kill 101, so we can keep being the money bucket for them to suck off of and use us at their will. Alright, anyway, so hunts them down, and eventually it is killed by the attack, and the team still isn't sure how this newly discovered bacteria reproduces and spreads to its next victim. So even scarier, it's transmissible. Now, how many of these insane diseases are we going to find that they're just discovering out of the blue are transmissible to other humans? Like, one we looked at was, we first started with like a BSE or mad cow disease, right? There was also that interspecies transfer, was like that blind cancer that we'd found that they knew about for years in milk. But they said, nah, it'll never spread to humans. Now they're finding it in breast cancer cells. So they're like, whoops, 30 years later since we've known about this viral entity in milk, and now it is passing to humans. Or Alzheimer's, the story we did about Alzheimer's potentially being contagious, not only through blood transfusions, but also through dental work. I mean, how many more of these things are we going to find that we've been at risk for and we never knew about them? Also, Zika, this new never-before-seen insane mutant Zika. Zika was just through mosquito bites, and all of a sudden it was through sexual contact, and then it was saliva. Holy shit, now it can be spread anywhere. And now there's the mysterious case we don't know how it got from one person to another. Maybe it's airborne. It's very frightening to think that here we are in the 21st century. We've had at least a good 100 years of improving technology. We are at a point now where it is beyond exponential, and we are about ready to take off into interstellar space with just some of this mad science gone mad. And they're just discovering bacteria and viruses, life forms that we've known about for probably 100 years. And we are just learning how transmissible they are after, literally that puts our medical technology of the last 30 years or so at a Stone Age level. If all these things, all these contagious biological organisms have been out there all this time and no one's been looking for them, and they've been able to pass from person to person, and they're causing disease, and we didn't know about it for all this time. Doesn't that make you scared? Doesn't that make you go, oh, how is it even possible? Don't they have microscopes and petri dishes? How would they not know all this stuff? Another thing I was thinking about, and I do have stories even queued up on some new research being done, prions. That was out of the BSE, the mad cow disease. They discovered a long time ago that not only are prions transmissible, not just between humans, but through the animal kingdom, and we also have the chronic wasting disease with deer, and there are some of the other animals in the animal kingdom that can get degenerative brain disorders, but we are also discovering that there's no way to kill prions, because prions aren't even alive. They're malformed proteins, and even scarier still, right? So we have all this science going on where they are just reconstructing biology in their own image and wanting to put it back into biological organisms to see what happens real time. And I don't know if we'll get to it today, but there is a brand new story about a new lunatic scientist who spent his research career on proteins, studying proteins, and being able to intentionally misfold proteins to create new therapies and drugs that now they want to start testing on humans. Now, you can see the damage the prions do. They make your brain look like Swiss cheese, and they have no way to kill them or stop them. So let's just give lunatic scientists carte blanche to start creating synthetic misfolded proteins, all in the name of medicine and the cure. Alright, anyway, let's get back to this new life form they found in living saliva. So previous research has shown that this bacteria contributes to gum disease, and when the new parasite is added, it seems to make it even better at evading white blood cells, which of course means that the body is less capable of fighting off the infection. So this is not only a new life form they're finding in saliva, but it seems to be pretty shifty and good at hiding, kind of like HIV and some of the other mysterious, quote-unquote, wink, mysterious diseases out there. The host bacterium also becomes more resistant to the antibiotic streptomycin, said the researchers, and now they'll be investigating the relationship further to try to figure out how the presence of this parasitic bacterium might affect human health and how we can use this knowledge to improve disease outcomes. While McLean and his colleagues have presented their findings to the annual meeting of the American Society for Microbiology, it has not been peer-reviewed as of yet, so we will wait for other researchers to confirm this discovery. But scientists are already pretty excited about it, rubbing their hands together, of course. Geobiologist Ronald Hatzpichler from where? Oh, the California Institute of Technology, who of course wasn't involved in the study, no, but he said that this could just be the tip of the iceberg for microbes like this. We might find out we've been infected by all kinds of new life forms that they've never seen before. Like, how insane is this? And it is unlikely to be the last discovery to come from saliva. As scientists think that as many as half of our microbes in our spit are to be properly identified. And this is incredibly exciting to see major advances in the study of lineages of life that until now have been impossible to cultivate. Gene data from other as yet uncultivated organisms suggest that host-parasite relationships between microbes are common in nature. So this type of study is a great template for others to follow. So now as we're looking at the story, I found another one that kind of connected dots with. This also is out of ScienceAlert.com. Scientists have developed a new technique that maps disease at a molecular level. Now, if they can map disease at a molecular level, then they should be able to also, create disease at a molecular level as well once they get that map. It is weird to think that with all the scientific advances we have had in 2016, and this is the story talking, but yes, haven't we just had amazing advances in 2016? Let's go to my Before the First Cup playlist on youtube.com/UCYTV and hit the Before the First Cup and just take a look at some of the titles and you're going to go, whoa, 2016 has been pretty insane. Anyway, with all the scientific advances we have had in 2016, like reusable rockets and the ability to clone dead dogs, and that is that cloning factory in China, we still don't really understand how most major diseases work when you get down to the molecular level. Ding! Now, I've said this so many times that as this scientific corporatocracy is taking over medicine and the government and lives of people, promising the dangling carrot of longevity and living forever and curing cancer, one of the things that they make us believe, or at least the general public, is that they know how to cure things. I've always believed My body was something that if I didn't take care of it, hey, it's just like a car. I'll put a new muffler on or whatever, but it's not like that. Doctors don't understand. They're like a computer technician, so when something goes wrong, they have maybe 4 or 5 things that they can try, but they're not really sure what the problem is, and it could be a combination of problems, and did you delete something or drop it? There's always some issues going on, and they can't fix disease like we were led to believe. They don't understand biology 100%, so they know exactly what's going on, though they will lead you to believe that they are the ones that understand the most when you bring up natural cures or any other type of alternative besides big pharma and taking medications to cure issues. But anyway, we really still don't understand how major diseases work when you get down to a molecular level. But Australian scientists have developed a new technique that can actually track the interaction and movement of proteins in a cell for the very first time, and could help researchers develop new treatments for what? Oh, cancer and other diseases. Now, it's almost sounding to me, I saw a while ago, a lot of the MIT publications come for a while happened to be about these new designer proteins that they were working on, and this is like the 3rd story that I've run across now in the last few days talking about them targeting proteins to be able to manipulate and control biology. This technique is called BiCAP, and this is the bimolecular complementation affinity purification. And what does it use? Oh, tiny nanoparticles to visualize and isolate protein complexes, in this case, breast cancer cells. Using BiCAP, the team can see when proteins interact with other proteins to form a complex and then track their movement. And it is important because proteins, in addition to being what your swole friends use to bulk up, we also are what make up the majority of our cellular processes in bodies. And if we can understand how proteins fit together and move around in a cell, it could help us better understand how they are involved in diseases such as cancer. These complex and highly choreographed protein networks are often disruptive in cancer and other diseases, says Darren Saunders from the University of New South Wales Faculty of Science, who led the study. But by mapping them, a bit like building a giant subway map, we can better understand the unique biology of cancer, and in turn, this helps us understand how cancer cells respond to drugs and may eventually help us find new targets for therapy. In an editorial in Science Signaling, which accompanies the new research paper, journal editor Nancy Go discusses exactly how important these molecular signaling proteins can be. The molecular side of signaling still has many secrets to reveal, and the importance of biomolecular interactions cannot be overstated. Although many protein-to-protein interactions have been studied, there remains a great deal to explore about binary protein interactions, much less multi-protein complexes. So right now they don't understand really how the majority of proteins interact within our body. So let's start messing with synthetic proteins and see what happens. I mean, they say the one thing on one hand, and they're like, don't worry about it. We got all these cures coming. Just step right up, you dead, dying, desperate, and we will sign you up for a free R&D to free up cash on our bottom line item budget. Make bazillions of dollars for the tech billionaire angel investors. And, do your duty. Do your duty being the real live guinea pig. Anyway, this BiCAP works by using an engineered nanobody, to recognize specific sets of protein fragments. And these fragments only form when 2 proteins have interacted, so it shows researchers when proteins have come together in a cell. While this technique has only been used in breast cancer cells for now, the researchers think that it could eventually be applied to all types of cancers, as well as other diseases. And the hope is that it will eventually help to discover new targets that could be used to fight disease, something which will hopefully lead to eventual new science and treatments. Now, let me see if I can find it. Here we go. Here's the protein science doctor titled "This Protein Designer Aims to Revolutionize Materials and Medicines." So now we have entire teams that are designing proteins, and even call them in the top picture unnatural proteins that they've designed. Dave Baker appreciates nature's masterpieces. He says, "This is my favorite spot," says the Seattle native, admiring the views from a terrace at the University of Washington. Hey, didn't we just see this is where some of that other science is going on, where they found that new life form? How interesting. And to the south rises Mount Rainier, a 4,400-meter glacier-draped volcano. To the west, the white-capped Olympic mountain range. But head inside to his lab, and it is quickly apparent that the computational biochemist is far from satisfied with what nature offers. Yes, another lunatic in the lab. And at least when it comes to molecules, on a low-slung coffee table lay 8 toy-sized 3D printed replicas of proteins. Some resemble rings, balls, and other tubers and cages, and none existed before Baker and his colleagues designed and built them. Over the last several years, with a big assist from the genomics and computer revolution, see, I'm telling you, exponential. This is sending us to the edges of biology, to now create life in our own insane image. With a big assist from genomics and computer revolutions, Baker's team has all but solved one of the biggest challenges now in modern science, and that being figuring out how long strings of amino acids fold up into 3D proteins that form the working machinery of life. Now, he and his colleagues have taken this ability and they've turned it around to design and then synthesize unnatural proteins intended to act as everything from medicines to material. Now, we thought nanobots were bad with little payloads, and then viruses with their guts sucked out and some sort of genetically modified cells put back into them and sent back into your body was then even worse. But now we're looking at unnatural proteins built to mimic nature, except modified and manipulated, and then wanting to be put back into you to treat disease. This is just beyond absolute science fiction insanity. I mean, we've seen stuff like this in the Bourne series of movies. We just watched one of the Bourne movies last night, and they were doing some sort of viral agent modification where they had to take the blue and the green pills, and really what they were these viral strings that would infect their cells, and then they had to be infected with the virus for it to lock in the genetic modification that it had done to them, whether making them smarter or faster or whatever. And I don't think that it's too far-flung, to be honest with you. You know, like I say, that even though this CRISPRing humans is some of the first genetic modification that we're being told about, you know, the DARPA has had their super soldier program and research going on for a long time, and don't forget that's behind a lot of locked, closed doors deep in the earth mountains in some unidentified place that research goes on. So, you know, anything goes there. You never know what they're doing. Anyway, back to the story. Already, this virtuoso protein making has yielded an experimental HIV vaccine, novel proteins that aim to combat all strains of influenza viruses simultaneously. Ding, 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 ding. Hold the phone. This almost sounds like it's involving our shape-shifting immune system here. I don't like this. You know, they were working on that HIV vaccine in a couple different places, so I don't know whose experiment that was doing the design of proteins, but I absolutely can tell you that universal flu vaccine was originally financed by DARPA and called the Blue Angel program back in 2006, and that was when they were messing around with the tobacco plant and putting genetically modified viruses and whatever they were making with their payloads and having tobacco suck it up and then taking the tobacco proteins and turning them into some sort of antigen that then they could put into a quote-unquote vaccine, which might not even be a stickum vaccine. It could even be an edible vaccine for all we know. Probably not with tobacco, but probably with the lettuce at Penn University. Lettuce is absolutely edible. They've had edible lettuce vaccines for quite a number of years already in other countries. But anyway, this is, it's yielded an experimental HIV vaccine, novel proteins that aim to combat all strains of influenza viruses simultaneously, carrier modules that can reprogram DNA cells, ding, 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 as well as new enzymes that help microbes suck carbon dioxide out of the atmosphere, oh my God, and convert it into chemicals. Now, I will guarantee you that that is part of the Department of Energy synthetic biology program that we have dug into as well. Now, what's the insanity in this? So let's suck carbon dioxide out of the atmosphere because, oh my God, carbon dioxide is global warming #1 gas, except that that's what plants thrive on. Remember, we read a story a while back that talks about, global warming and carbon dioxide will cause more plants and trees to grow. But hey, trees and plants are responsible for more global warming. Do you remember that? I mean, what idiot science is that? You're not going to tell me that the more plants we have, the hotter it's going to make the planet. Because I beg to differ. I want to see the numbers on that. I want to see somebody prove to me that by having more green and more shade from the sun, that the planet is going to heat up more. Because I call absolute and utter bullshit on that. Yeah, let's kill all the trees. Let's suck all the CO2 out of the atmosphere, make us safe. And if you remember, Bill Gates, again, at that TED conference where he talked about using vaccines to reduce populations and how much easier it'll be with a lower population to deal with problems, he also talked about, when he showed that equation, bringing the carbon quotient down to 0. How interesting now. The microbes that suck carbon dioxide out of the atmosphere and then make them into something that they can make bazillions of dollars at without paying back to society. It's great. Baker's team and the collaborators report making cages that assemble themselves from as many as 120 designer proteins. You know when they've been working on that? MIT's Weiss Institute for Biologically Inspired Engineering. Yes, that was those little DNA cages, which could open the door to a whole new generation of molecular machines. And that's where we saw the father of nanotechnology, good old Eric Drexler, at MIT, talking about molecular machines. That would be the day that all manufacturing would change from a manufacturing plant to desktop manufacturing and being able to create the first generation of the Star Trek quote-unquote replicator, to be able to actually create matter from the building blocks of nature. What insanity is this? If the ability to read and write DNA spawned the revolution of molecular biology, the ability to design novel proteins could transform just about everything else. You better start paying attention, guys. Nobody knows the implications because it has the potential to impact dozens of different disciplines, says John Molt, a protein folding expert at the University of Maryland's College Park. He says it is going to be totally revolutionary. Now remember, I pointed out to you guys how, we used to have once in a decade, we would have a technology that was considered quote-unquote disruptive. And disruptive technologies are dangerous because they take everything we know and love. They take things that work in the economy, that people's entire livelihoods and educations, and turns it on its head and throws it out a window. It gets rid of entire industries and leaves us with a big problem. But typically, that's been, in my lifetime, every decade or so, that's been happening. But with the law of exponential returns, and that's what we are experiencing right now, and fast forward with all of this supercomputing, and artificial intelligence, and improvements in being able to visualize components at a biological and electronic level, be able to make components at a nano scale right now that then can develop other components at a nano scale. They have self-assembling capabilities and program logic into biology just as if it were some sort of electronics. We have reached a point now where this year alone, 2016, there have to be 10, 15 disruptive technologies that have all hit the stage. You know what that means? That means that everything, absolutely everything that we understand today as our reality, very shortly in the future will no longer be our reality. And I think that's why every day feels like science fiction over the last couple years, because it really does. You know, the material that I use to do my show, I know it sounds like lunacy, but it's not. This is like science fiction meets reality with a little touch of evil and a double-edged sword, using dangling carrots to suck you in 3D virtual reality goggles so you're not paying attention. And this is just beyond belief. But it is actually happening. And they will be able to manipulate and control every bit of our reality. I saw a story yesterday, and I don't remember exactly what it was, but it was about Google, some Google venture, where this new technology that they're working on will literally be able to build reality from scratch, they said. Reality from scratch. And what does that mean? We'll be hyperdimensional somewhere living. And I mean, we literally, right now, we had Chinese or Japanese scientists created touchable holograms, people. Touchable holograms. Light and matter can exist in the same space at the same time. They've been able, with quantum and some quantum experimentation, to actually utilize quantum entanglement, the existence of matter at two locations at the same time, and use them to communicate over extended distances, like mental telepathy type stuff. I mean, this is just, and it's so beyond belief that the majority of the people that you would try to explain any of the science to, or some of the things that are going on, or some of the things that are going to hit us in the face, of course they're going to spin all this mad science into some consumeristic product, like a dangling carrot that'll make you want to buy it and become part of this machine, open your arms and accept it, because it'll make your life easier, or make your life better, or make nano longer. But in reality, it is a complete and utter takeover of all life on this planet. Somebody's going to hold all those keys for that synthetic protein that they developed and got inside of you. Somebody else is going to have total and utmost control over what happens to that protein after it's been inside of you. I don't think they should be trusted. I don't think any of this should be trusted. Tread lightly, because on one hand they tell you, oh, we're the scientists, we know everything. Hey, this is going to be good for you. This is mandatory vaccines. Step right up, little girls. Get your Gardasil with the virus-like synthetic particle loaded with our little genetically modified payload we want to put in your 11-year-old that hasn't really been tested over multi-generations. And oh, by the way, did I or did I not just see a story in the American Journal of Pediatrics out of January 2016 that says, yeah, well, you know what? Some of you lunatics that said that you had these little girls going into premature menopause at like 11 and blamed it on this vaccine, you know, you might be right. There's been 3 or 4 documented cases now of little girls going into menopause. And you know, there's something that when I first, my doctor, my pediatrician, who's telling me all about this Gardasil vaccine and how it's highly recommended and how angry she would be if I didn't get my daughter protected against cervical cancers and all these vaccines that will do this and take care of her. You know, I asked her a couple things about that, and I told her about that scientific case study that I read out of England of that little girl who had ended up going into menopause, and she told me that did not exist, it never happened. When I got back from that appointment, I found that case study. I went to the website, I typed in a little message box that my doctor asked me to send her the article about the little girl who went into menopause, and please forward it to her. So I will guarantee you, they probably didn't give it to her, but I got now fodder to throw in my doctor's face. This is going to tell you they don't know everything. This woman's recommending this shit to me, and she obviously did not do her research. If I found a documented case of some real issues, that maybe I'd want to proceed with a little bit of caution instead of going with the marketing literature that they're spinning on TV. Yet this doctor did not even mention it to me, and looked at me like I was a tinfoil hat wearing lunatic and told me it never happened when I mentioned it to her. Then we also found out the Gardasil #1 vaccine, after they released it and it was mainstream news, and whoops, we discovered that very first vaccine that all my friends gave their daughters actually made those children more susceptible to those 8 cancers that they were supposed to protect them from. But don't worry, because we have a brand new Gardasil 9 vaccine that not only will fix our first issues, we put some new protein in there, but it'll protect you against more cancers as well, so you better step right up and get another dosage. The disturbing fact about that is if you read the insert under side effects, and I think we read that on the show that day that we did that, it was like death and all these other cancer. And none of these vaccines, and it'll say it right in the insert, have ever been tested for mutagenesis. Ever. Now, mutagenesis being cancer. Why would you inject your child with some sort of mad science gone mad vaccine with all kinds of synthetic shit in it that causes worse cancers than if they didn't get it at all, with something that has never been tested for mutagenesis? I think that right there should be the rubber stamp for everyone to say no. And that was just in the Gardasil vaccine. Let's go through all the other inserts and take a look at what other lunacy is in there. I mean, we know about all the recombatant proteins and DNA and monkey viruses, FV40, bovine serum out of cows. I saw in a CDC document, too, that they mentioned in a document that it could potentially spread bovine spongiform encephalopathy if the animal had been infected. Didn't they ever test for that? And guys, this is absolute lunacy. You can trace it all back to Plum Island. And before that, you can trace it all back to Operation Paperclip and German mad science. We brought all those guys over here, and the humans became the product. We became the guinea pig. Those programs never stopped. DARPA kind of got spun off because it has to be part of the military, because it's all about national security. So anyway, here we go. The ability to read DNA spawned the revolution of molecular biology, and the ability to design novel proteins could transform just about everything else. So look out, people. The machinery for building proteins is essential for all life on Earth. And so if we can build proteins, hey, we can control life now. Isn't that ergo? One way around the problem is to determine protein structures experimentally through such methods such as X-ray crystallography. Hey, one of my very best friends is an X-ray crystallographer at a university. And nuclear magnetic resonance spectroscopy. But that's even slow and expensive. And even today, our protein databank, which is an international repository, holds the structures of only 110,000 proteins out of the hundreds of millions of more that are thought to exist. So I don't know, guys. I don't really know what to say about all this, except that we better put on a seatbelt, because they'll be able to create a protein for every purpose it says. The ability to predict how an amino acid sequence will fold, and hence how the protein will function, opens the way to designing novel proteins that can catalyze specific chemical reactions or act as medicines or materials, and genes for these proteins can be synthesized and inserted into a microbe, which then builds the proteins. Oh, jeez. Here we go. This is exactly the stuff we've been talking about, and this is like the cherry on top, the big, scary, rotten cherry. Baker's Lab is abuzz with other projects, and last year's group and collaborators reported engineering into bacteria a completely new metabolic pathway, complete with a designer protein that enables the microbes to convert atmospheric carbon dioxide into fuels and chemicals. 2 years ago, they unveiled in Science protein that spontaneously arranged themselves into a flat layer like interlocking tiles on a bathroom floor. Yeah, those little octagons. I I saw these stories, and such surfaces may lead to novel types of solar cells and other electronic devices. Yeah, electronic devices made with biological synthetic material. In perhaps the most thought-provoking project, Baker's team has designed proteins to carry information, like a computer, imitating the way the DNA's 4 nucleic acid letters bind and entwine in the genetic molecule's famed double helix, so create life from scratch. But now, hey, don't worry about it. These protein helixes cannot convey genetic information that cells can read, but they symbolize something profound. Protein designers have shed nature's constraints. Yes, get rid of Mother Nature. We're gods now. And are now only limited by their imagination. And hey, I'm not into minotaurs running around or any other scary creatures. He says we can now build a whole new world. Yes, it's all about the new world order of functional proteins. Be afraid, be very afraid, people. This is the type of stuff that George Church was talking about. This was the type of stuff that was topic of that Harvard secret meeting of 150 geneticists talking about creating synthetic DNA and synthetic life. It should be frightening to the futures of all life on this planet. Now, if not to add insult to injury, we're going to finish up the show looking at milk, since milk is the manna of early life. There was a new discovery. Scientists have now created milk that lasts for 9 weeks without going bad. 9-week-old milk makes you thirsty. You might not have to worry about milk going off in the future. Oh, the future is going to be so grand, with scientists announcing the development of a new technique that can keep it unspoiled for up to 9 weeks. With an add-on to the regular pasteurization process, the milk's temperature was increased by 10 degrees Celsius for just 1 second. And that added up to 7 weeks to the usual lifespan of refrigerated, pasteurized milk. This new process, developed by researchers at Purdue University in Indiana and the University of Tennessee, eliminates more than 99% of the bacteria left after pasteurization, and with the treatment, you take almost everything out. So, let's just kill everything. Absolutely all the good bacteria. Anything that is in that milk, let's kill it all. Hey, that's what we do best, is kill. And with this treatment, you're taking almost everything out, says lead researcher Bruce Applegate from Purdue. But what does survive is at such a low level that it takes much, much longer for it to multiply to a point at which it damages the quality of the milk. You might already be familiar with pasteurization. Yes, pasteurization that kills all live bacteria. Invented and named after Louis Pasteur, where the milk is heated to kill off pathogenic bacteria which would otherwise make us ill, or which would otherwise be available in raw milk. That seems to be quite healthy for you. Putting milk in the fridge helps to temporarily contain the stubborn bacteria the pasteurization leaves behind, but of course the milk does eventually go off. Thanks to an extra burst of heat in this technique, that safe drinking window now stays open for longer. So, it makes me happy that at least they're not putting some genetically engineered bacteria in there to keep the natural bacteria in check, but on the other hand, when they start manipulating our food supply and saying, look, it's just like the old stuff, just better, you always worry, because they don't need to label any of this stuff. They're going to put some kitschy marketing shit on it and tell you it's new and improved. Probably increase the price up $10 or something. This is all going to be in the name of climate change somehow, Keep you happier and healthier. They're going to start infusing nutraceuticals into your milk, too. And yeah, I can see that happening in the not-too-distant future. But it's got to make you wonder, because it sets the normalcy bar for you to be comfortable with them manipulating your food in ways that didn't naturally occur. We're already comfortable with things. They started radiating meat and vegetables and spices a long time ago. No one really complains about it. They can vacuum pack them and send you meat from China and you never even know it. But hey, it's cheap. Let's just go to Walmart and do our grocery shopping. Alright. Anyway, and last but not least, to stay on the vein of milk, and this is where we actually ended the show yesterday as well. All in the name of climate change, because like we said, cows cause too much methane. We need to find some alternate sources of protein in the future. And with some sort of 3D printed nutraceutical shaped in the shape of good old Mickey Mouse ears to make the kids want to eat them and love them too. Scientists now think good old edible bug protein, the cricket protein has been already on the map for a while by the UN. Scientists now think that cockroach milk could be the superfood of the future, so don't worry about it. We're already messing with your milk. Set a little bit of normalcy there so you don't care about it. Well, hey, one of these days it might not be cow milk. It doesn't say cow on the package, does it? It just says milk. An international team of scientists has just sequenced a protein crystal located in the midgut of cockroaches. The reason? Well, it is more than 4 times more nutritious than cow's milk. And the researchers think it could be the key to feeding our blossoming, exploding population of the future. So let them eat cricket protein and cockroach milk, and it'll be healthy for all of you. Although most cockroaches don't actually produce milk, we're going to have little cockroach milking machines. The Diploptera punctate, which is the only known cockroach to give birth to live young, has been shown to pump out a type of milk containing protein crystals to feed its babies. And the fact that an insect produces milk is pretty fascinating, but what fascinated researchers more is the fact a single set of these protein crystals contains more than 3 times the amount of energy found in an equivalent amount of buffalo milk, which is also higher in calories than dairy milk. Clearly, milking a cockroach isn't the most feasible option. Well, but an international team of scientists headed by researchers at the Institute of Stem Cell Biology and Regenerative Medicine, and our favorite mad science country, India, has decided to sequence the genes responsible for producing the milk protein crystals to see if they can somehow replicate the cockroach milk in the lab. Oh, Jesus. So not only do we have China making genetically modified cloned cows with human breast milk genes in the cow to produce human milk, but now we got the Indians producing, replicating synthetic cockroach milk in the lab. And don't worry, because these cockroach milk crystals are a complete superfood. They have protein, fats, and sugars, and if you look into the protein sequences, they have all the essential amino acids, says Sanchiri Banjari, one of the team, in an interview with the Times of India. Alright guys, well I think we're going to leave it at that. Make sure you stick around for all kinds of great new radio. You have an awesome Thursday, and I'll catch up with you tomorrow. Everybody take care.